Yeah, many thanks. I really try to adhere to the heights you just put me up onto. Just a bit of context before we actually start. You might wonder why there is actually a spider on my screen. That's surprise, surprise. I'm a spider guy. Here we go. So yeah, I'm a spider guy. However, I'm setting up my own lab in Gießen right now, and we are a very cool bunch of people. And this image is not really up to date anymore. We have grown tremendously since. Now we are almost twice the size. And we work with all kinds of different stuff since. So we have a couple of snake projects running now. We also used to work on amphibians. However, the most work we do really revolves around arthropods, also mostly on spiders. And this brings a couple of particularities with it. In contrast to what most of you may be used to, we have a few problems when we work with our venomous animals, because the traditional workflow that most people apply in venom research is really the traditional one where you start at your organism, you collect the venom, you do some chromatography, and then for each fraction you study the activity and the structure function. And then in the end, you derive your knowledge, which in our case usually is the question, can we use a specific toxin as a drug lead or not? This works pretty well when you work with venomous reptiles, such as snakes, or here in this case, an almost snake, so to say. However, if you work with smaller arthropods, as we do most of the time, this is a bit limited, because this whole workflow based on chromatography really is sample intensive, and we need a couple of milligrams usually to do that, which is almost impossible to get from those guys. These things have almost no venom yield, and usually we have to make hundreds of thousands of specimens to even do a single chromatography run. Because this is really inconvenient, we really switched methods here a bit. We, instead of doing this traditional workflow, we completely switched to doing modern Venomics, which we have heard this morning from Mike and Cassandra already. So we do a lot of venom proteomics of milked crude venom, and in the specimens where we cannot milk the venom in the first place, we usually extract RNA from venom glands and sequence the venom gland transcriptome, and if possible, we actually combine those workflows as displayed by Cassie just in the last lecture. This methods combination allows us really to disentangle the venom composition of almost any animal, no matter the size. And once we have our venom composition unveiled, we on top of that usually apply different in silico methods to identify potential drug candidates from there that then are interesting for our downstream research. However, this combination does not really solve the fundamental problem that we have, because Venomics is great, it helps to unveil venom compositions, but it doesn't provide us access to the components themselves in the lab, right? So this is how biotechnology came into my life, because we are thinking about that, and we, instead of trying to extract these molecules from these tiny amounts of venom, which would be extremely tedious, we thought that it would be really smart to just use the sequences that we have identified anyways, and just set them into plasmids, clone them, and express them in E. coli, or also even use cell-free technologies where we make artificial cytoplasms, and then we just create our toxins in a couple of weeks or days in a lab scale, and then from there we can proceed to subsequent functional assays. And this is really how we went into biotechnology, and thereby we kind of fell in love with the whole thing. And it's not only we, actually there are now many groups around the world that focus on different aspects of biotechnology and apply it very differently to unveil the secrets of venom systems. And I think that this whole connection of biotechnology protocols and tools is really the next thing on top of the ongoing in silico and omics revolution that we see in Venomics right now. And what I want to do in my talk here, I don't have that much time, and each of the methods themselves would be an independent lecture. But what I want to do is to give you somewhat of a bird's eye perspective on what is currently available for venom biotechnology tools, and show you a bit what is doable, what may come up in the future, and to give you a feeling of what we really can do and which questions can be answered. And for that, I want to go on over the different methods based on the traditional hierarchy of genetic complexity, starting on the genome level, looking at where biotechnology can be used to study the genomic blueprint of venom systems and answering questions about venom system development, venom gene regulation, also modes of action. From there, we slowly proceed into the level of the gene product, which is the toxins themselves, looking in structure-function relationships, how we can use it to access non-milkable taxa, which is for me the most interesting application, but also their artificial derivatives could be created with the same technology. And after that, we quickly summarize how biotechnology tools can be used to study venom gene function, which means unveiling modes of action and looking into exochemistry, but also studying venom resistance and again, diving into structure-function relationships. Well, the first thing I wanted to talk about is how we can use biotechnology to study the genomic blueprint. And the most important method for that is what we call functional genomics. So tools that we can use to actually manipulate the genome itself, or to at least engineer the gene expression of your model system. And here we have two major methods at play. The first one is RNA interference. The second one is, you should know it, CRISPR-Cas9 screenings. Both are fundamentally similar because they work on the nucleic acids. However, the mechanisms are a bit different. In RNA interference, you essentially create a short piece of RNA that is homologous to an mRNA in your cell, and you inject it into your system, and you trigger a chain reaction, so to say, between proteins in the system and these mRNA that cause the subsequent degradation of the resulting mRNA. So in a nutshell, you make a specific piece of RNA and you kill all the mRNAs that are homologous to it in the cell, which causes a transient prevention of gene expression of the gene that is encoded by this mRNA. So you are enabled to do lots of function studies, and that both in vivo and in vitro. On the other hand, we have CRISPR-Cas9, which is fairly similar in that you can remove gene activity, but here we have actually stable knock-ins or knockouts. So you have really a genetic scissor, so to say, that is used to excise parts of the genome, and then you can also engineer it to add novel genes into it. This also works in vivo and in vitro, and as I said already, it enables loss of function plus gain of function studies, and they both are extremely useful. How they can use, the best example of that is a recent study published, I think, in 2018 or 19, not on a snake, unfortunately, but on a very interesting venomous animal, which is Chironex fleckeri, the box jellyfish. And this study used gecko screens. So the gecko screen essentially is a system where you use your cells and you create a cell colony where you statistically have knocked out each gene of the whole genome across the whole petri dish. Then you copy plate it and use those copies and select them with venom and compare them with non-selected cells over time. Of both experiments, you harvest the genomic DNA, and then you can amplify the leftovers of the CRISPR-Cas knockout of each gene. You can compare these fingerprints, so to say, and this helps you to really identify the genes that are responsible for venom susceptibility and resistance. And it is a bit complex. However, if you use it properly, and the authors did that very well, you can use this approach to really disentangle and dissect the mode of action of the whole venom cocktail on the cellular level, and you can come up with these very nice pathways and interaction models of which genes are involved on the cellular level to make the cell actually susceptible to venom. And this can, of course, then downstream be used to create novel antidotes and therapies. And also it can be combined very well with mouse models and also with histological sections to undermine the hypothesis generated by the screening in the resulting models, so to say. To employ functional genomics, two major considerations need to be kept in mind. The first thing is you actually need to have high quality genomes, because the genome is really where you derive your sequence for manipulating the whole system in the first place from. Luckily, we have been given the gift of many high quality genomes that have been published over time. I cannot even mention all of them on a single slide, but when I started Venomics a couple of years ago, there was almost nothing except the king cobra. And now slowly or steadily, even more and more genomes are emerging, particularly from snakes. So this is no longer really the obstacle anymore, I think. But there's a second requirement we need to have in mind, and that is we need to have the ability to keep the whole thing in cell culture. So we need to have cell cultures, or at least manipulatable models to really employ the functional genomics. And this is where the whole thing becomes a bit messy and difficult, or at least it used to be that. We talked about the importance of venom collection earlier today, and that this can be actually be a bit tedious, right? This is partly because it again has a few obstacles to be overcome. So venom collection by milking, it requires you to do field work, and if you are not going to do field work, you at least need to have access to captive animals to actually collect the venom from. In many specimens, in my arthropods to the extreme, but also in some snakes, you have a very limited yield. But for experimental considerations, even more important, especially wild-caught specimens, this is not a controlled environment, right? You have no idea what the animal did the last days. This is a very, random sample, so to say. And also, wild living animals are very difficult to manipulate. Another thing that we have just talked about in the last session is also bureaucratic obstacles. In Germany, at least, every venom collection is considered an animal experience, is linked to tremendous amounts of paperwork and bureaucratic obstacles that you need to overcome. So with that in mind, how cool would it be if we would have standardized and manipulatable venom systems where you can do experiments on, right? This would be really amazing. And luckily, we also talked about that earlier briefly, is we now have that. We now have organoids. What is an organoid? An organoid is essentially an artificial mini organ that is an in vitro miniaturized version and simplified version of a natural organ that can be kept in cell culture. For instance, here we have a selection of colon organoids at different development stages. And here a final organoid really is a thick body of bioactive cells. They are created by taking biopsies, and the created biopsy is then either embedded into a system and turned into tissue fragments, or single adult stem cells are taken, and these then can be kept in cell culture, and with the addition of different growth factors can be differentiated in all kinds of different tissues to form organoids. And just a couple of years ago, luckily, the first study describing the establishment of venom gland, snake venom gland organoids, has been published. As I said earlier, the study started by extracting venom gland tissue from embryonal snakes. In this case, it was Cape coral cobras, Aspidelaps lubricus. And these venom glands that were dissected were then digested into tiny cell fragments, and from there, over different stages and addition of different growth factors, really biosynthetically active organoids were created. It's really interesting to follow the development of the organoids from being tiny fragments of cellular debris in the beginning after creation, over the development towards really round and nicely agglomerating, apparently biosynthetic organoids. So you see there is a really nice development in there. Even when you compare the structure of the organoids with that of the venom gland sample, you see that the venom gland organoids phenotypically somewhat already resemble actual living snake venom glands, which is to me really astonishing. We have now seen that the organoids actually have a very similar histological architecture as venom glands, but the question is, are they also similar biology-wise? Do they actually function in a similar manner? And this has been investigated as well. We have just learned about transcriptomics, and that was applied here. So RNA was extracted, a venom gland organoid transcriptome was assembled, and then the resulting fragments were compared for identity and also expression. And this revealed that actually, indeed, the expression profile of venom gland organoids is fairly different to that of most body tissues. But most interesting, when you look onto the actual venom mRNA profile, you see that in comparison of the venom gland organoid here, the mature one, in comparison to the venom gland transcriptome of the actual venom gland, you see it's fairly similar. You have an extreme high overlap of 3F3X toxins and also of the CRISP toxins. The fingerprint is fairly near natural. So apparently organoids really resemble somewhat natural mimics of venom glands and therefore can probably be used as very powerful model systems to study the foundations of gene expression, the regulation, and all of that. Another important consideration to be made when looking at organoids, but also in cell culture systems in general, is their longevity, because a short-lived cell culture is not very useful if you have to reestablish it every week. Luckily, this has also been investigated quite in depth already. Quite a bunch of different snake venom systems have been used to create venom gland organoids, both from embryonal and adults. And you see here that actually they are quite long living. The organoid system based on Aspidaps lubricus was successfully cultured for almost 2 years. The other ones at least half a year to 3 months or a month. So apparently many snake venom gland organoid systems are quite long living. And though they can also be used for a wide range of applications. They have been used for fluorescence activated cell sorting, of course, RNA extraction, fluorescence and antibody experiment, cryopreservation, so biobanking, but also to harvest toxins. And this really shows that organoids are a very promising system for the future. However, non-snake venom organoids are not yet established, but I know for a fact that several groups are on that, and I expect arthropod and even mollusk venom gland organoids to be popping up soon at some point. With that, I want to proceed to the second part, the level of gene products. And for that, I want to briefly talk about what I have done earlier, this traditional workflow where we have to overcome this bottleneck of chromatography because the many small specimens, including also some rear-fanged snakes, certainly don't deliver promising venom yields, and therefore this traditional workflow is kind of limited. And I also already said that in my group, we overcome this mostly by applying heterologous expression in E. coli to allow functional tests downstream. And heterologous expression really relies mostly on E. coli. Several other bacterial systems are also available, but E. coli by far is the one that is easiest to handle in the lab. Most protocols are established for this bacterium specifically, and it really is the working horse, not only in toxin expression, but I would say in most biotech applications globally is probably the most important one. If you look on expression of toxins, we have 4 major aspects to or different types of expression systems at hand. The first one is production as inclusion bodies, where you create in the cytosol agglomerated, usually misfolded agglomerates of the desired toxins. These can then be gained from the cell by lysis and need to be processed to actually yield the folded protein in high yields. This sounds difficult, but actually in large-scale application, thinking of industrial workflows, this is actually how most industrial production pipelines work anyways. So if you want to use a toxin for pharmaceutical production, this is actually the way how you want to create your expression system. It's for you in the lab scale much more difficult to do, but for the final application, this is actually desired. When you look on lab scale expression, you usually use periplasmic expression. That is, you express your desired protein in the periplasma of E. coli, and the reductive environment supports protein folding into disulfide bond formation, but you also have the ability to express, similarly to your protein in a co-expression way, several enzymes that support folding and disulfide bond formation, but also you have engineered strains that lack proteins that affect the folding and disulfide bond formation in a negative way. So all kinds of different modifications can be made to the E. coli system in order to achieve expression of your protein of interest. When you express toxins in E. coli, this is usually done as fusion proteins. Fusion proteins, as the name implies it, are a fusion construct based on a fusion partner. Different kinds of proteins are available for that. I'm going to talk about that just in a minute. That is linked to your toxin of interest. And in this state, because of the large size of the fusion partner and stereo chemical interactions with the active side of the toxin, usually this construct is not bioactive, and that is very useful because you don't want to create a bacterium that makes a type N toxin, and then you get lost it somewhere in the environment. That would be not very useful. So you want to have something that, you know, doesn't kill anyone when it's set free. That's why we use the fusion partner system. But the fusion system also has different applications on top of it. Several fusion partners support the expression of the protein, help in purification, they support the folding, and also help in detection. Once this fusion protein is synthesized by the bacterium, you usually yield it and enzymatically cleave off the active toxin from the fusion partner. This one is then going to the trash bin, and the toxin of interest is then forwarded to our functional assays. A wide variety of fusion partners exist specifically for E. coli. I'm not going to talk too much about them, but the Sumo one and also thioredoxin you should know, because these are, at least in my experience, those that work best and are also quite often used. Another very cool fusion partner is GFP from the jellyfish Aquariora victoria, which is a green fluorescent protein. This, although I don't have personal experience with it, it is fluorescent, so it can be used to actually track your construct. And this may be for different experimental setups be of interest, I think. Besides E. coli, there are also different other methods for protein expression. Eukaryote one specifically. We have a yeast system, usually it's Pichia pastoris. Insect cells are available, mammalian cells are also available. They have the advantage over E. coli cells that they are able to do more complex post-translational modifications, glycotrees, phosphorylation, you name it. They are also usually, because they have a proper compartmentation system, they are better in folding. However, they are also usually much more expensive to maintain, more difficult to manipulate, and they are overall much less commonly used. So most studies still use E. coli systems. However, I think that several eukaryote systems also will be seen more often because of the ability to do these modifications, which in case of snake toxins very often is important. Another interesting system are cell-free systems, and these are particularly cool because in a cell-free system you essentially use your Eppendorf tube, you fill it with a cell culture extract of your desired expression strain, for instance in E. coli. It contains all parts of the genetic expression machinery, so it contains the ribosomes, the chaperones, all that is needed to make proteins. You add your reaction mixture with all the D-entities and all the cofactors to it, and then you can also just add a gene construct, which can be a linear gene fragment, but it also can be a plasmid. And by mixing all that into an Eppendorf tube, you essentially create an artificial cytoplasm in your tube. This can be cultured then overnight, and then all the protein biosynthesis is happening in that tube, and you can actually harvest the toxin from that as well. This is a very cool method because it promises to be very quick. You circumvent a lot of the different parts of the GMO creation pipeline that are sometimes a problem. And therefore, we looked into that very recently. We dabbled in that and compared different available systems in a spider venom toxin. And what we found is that of the 3 systems we tested, however, only one was able to do the protein, but we were able to actually measure a bit the activity, but the yield was very low, and we also saw problems in protein folding. However, for chemically extremely complex spider toxins, this whole cell-free expression may be of limited use, but there have been already other people working on it, and at least for snake venom, a calicrein has been created in a wheat germ system, and it actually also was bioactive. So although this is an emerging technology, it has not often been used in context of toxinology. It is apparently powerful, and again, I expect it to become more prevalently used, particularly because very recently the people around Glen King in Australia really engineered a cell-free expression system that is really well adapted for the expression of extremely chemically complex toxins. They successfully expressed several ICK toxins of spiders, but also cyclic peptides from plants and all that. And I think based on these developments, I'm confident that cell-free expression will be seen more often in the future. Briefly looking at the differences in the expression protocols and their advantages and disadvantages. in E. coli system, we transform our bacterium, we grow it. At some point, we harvest the toxin then by cell lysis, and then the fusion partner is cleaved off and purified, and then in the end, we have the toxin after chemical purification. In the cell-free system, however, we just culture a bacterium, we grow it, we lyse it. The cell lysate is then used to be the basis of the cell-free system. We add our fragment and express in the Eppendorf tube and purify them again. If you go with commercial systems, all that is skipped, and you immediately buy a finished Eppendorf tube with everything inside. You just need to have to add your gene fragment. The heterologous system contains multiple steps. It has a disadvantage that a GMO is created. However, you have a wide range of hosts, and therefore you are very flexible with that. The cell-free expression systems only contains very few steps. Very often, if you go for commercial ones, even it's a one-step thing. You don't create a GMO. However, you have a very limited variety of hosts, but systems for pro and eukaryotes are available. However, cell-free systems have problems with yield and folding. The last thing I want to talk about is the last level, the gene function. Once we have expressed our protein, we want to study it. And here also, biotechnology really advances on what is doable, particularly in the area of biosensors. So what is a biosensor? A biosensor is any system where you use a biomolecule that reacts with your analyte, and the device measures the chemical reaction in some form and transfers it into an electronic signal that can be measured. Biomolecules used often for that are antibodies, enzymes, whole cells, aptamers, or even nanoparticles. And there's also a wide range of transducer systems that actually convert the biochemical signal into an electrical one. The most commonly used system of biotechnology to study toxin function actually are xenopus oocytes and patch clamp electrophysiology. However, most people always forget that this is actually a biotechnology approach. In this system, you take eggs of the African claw frog, Xenopus laevis, and you express ion channels and receptors of your interest in it, similar as you would do express a protein in E. coli. You can also transplant receptors from one biomembrane into the Xenopus oocytes, and this artificial oocyte is then attached to a system. It is patched, and this system measures the activity of the expressed ion channel. It measures the flow of ions, the current, and transverses that into an electrical signal again, and allows you to really measure the effect of specific toxins on a selected ion channel. Different technologies emerged to do patch clamp with Xenopus oocyte. We have cell-attached, whole cell, outside out, and inside out protocols, and all these different patch clamp technologies have been used to study a wide range of neuronal receptors, including natrium channels, potassium, calcium, acetylcholine, and also GABA channels. And if you want to learn more about that, there is a very recent, brilliant review on that whole thing published very recently. The last technology I want to mention is biolayer interferometry, which is another very fancy but upcoming technology that we see very often now. This whole thing, although it has a very complicated name that challenges my list a lot, works by measuring the interaction of an immobilized protein with its analyte and the resulting changes in optical interference. So you have a molecule, it is attached to your measuring surface. You have an incident white light that is going through, and once the analyte is binding to it, you cause variation in this interference, and this can then be measured. The cool thing is that now we have the opportunity to actually engineer these bound molecules quite a lot. And this is thanks to mimotopes. A mimotope is an artificial peptide that mimics an epitope, and it has similar biochemical interactions as this epitope. And these mimotopes can be bound to BLE systems to measure interaction with almost any protein. Here's an example, very recent, of the alpha-1 autosteric binding site and its interaction of different neurotoxins, alpha neurotoxins interacting it. And the mimotopes here were created as mimics of the binding sites of fish, amphibians, lizards, snake, bird, marsupials, and rodents, and they were attached to the BLE system and confronted with the neurotoxins, and then the activity of this mimotope with a specific toxin can be measured. So mimotopes in combination with BLE systems allow us to create custom-made substrates for all kinds of targets. And that is what I think is really cool. And it has been employed pretty widely in the last years, particularly the group from Brian Fry is pioneering that, I would say. We have found neurotoxicity in beetles with that. The system was used to unveil a novel mechanism of electrostatic resistance in different receptors, and also to study the venom resistance in Afro-Asian primates. And I think, again, this is a very powerful technology. The last thing, and then I conclude, I promise that, is I want to highlight that it's not the individual technology that is now available to actually study venom systems. Each of them is incredibly powerful in their own right. What makes them even more disruptive is that they actually create, in a synergistic manner, these experimental positive feedback loops where you can merge all these different technologies. They are interfering and build upon each other to create very in-depth studies that are not doable without them. And most importantly, they also resonate quite well with Venomics. So it's not a single technology, it's not Venomics, it's not Venom Biotech, it's the whole package used in combination with each other in a very smart way that is really powerful. And I think this combination of methods brought us as toxinologists now to the point that it's actually our own creativity that limits the experimental design. And I think that is really awesome. With that, I'm just in time. Thank you.